So yeah, I'm going to talk a little bit different. So switching more to cell biology. So I hope I can make everything clear for you clinicians, physiotherapists, more mathematical people. If not, feel free to interrupt me. We're going to talk a little bit about the research we're doing in Leiden. Then we're going to talk a little bit actually about our organoid model. So that's the inner ear and the dish we're working with, and how we actually use it also to study disease. So when we genetically modify them, how does this inner ear organoid model change? And lastly, we're going to talk a little bit about how we actually improved this organoid model a little bit recently. So first of all, our research group we call Autobiology Leiden, and we have like a really broad aim, and it's actually to address the burden of hearing loss and balance disorders. And we're doing this in two different ways. And the first way is really to understand human developmental biology of the inner ear. So what's normal development, but also what goes wrong when pathologies arise. And secondly, we're working actually towards new therapeutic strategies. And also have an example of this in this presentation. So if we look really broad at studying in the ear and disease, we can of course do a lot in animal models. We can genetically modify them, we can treat them with different compounds, we can rotate the vesicle and see what happens to the development. But that's actually a stark contrast in what we can do in humans, because these studies are really more descriptive. So a lot of these electron microscopy studies from the 80s, but also more recent studies with immunochemistry, just looking at what happens to the human inner ear. And that's actually where we think we can contribute to, and that's with in vitro models of the human inner ear. And so that's the organoid model we use. But if you look in literature, there are some other models also out there that actually is just human inner ear tissue in a dish. So here on the left, you can see a paper from Marta Rossio, in which she collected fetal inner ears. She dissociated them, collected cells from the prosensory duct floor of the cochlea, so that's the development organ of Corti, and brought this back into culture. And they form like sphere-like structures, and providing the right medium and also providing some mesenchymal cells. They actually also develop hair cells. So this is a human inner ear model in a dish. But it's not really high throughput because you need this fetal inner ear tissue. Then also work from our group is actually that we collect also fetal inner ears, but also utricles, for instance, from vestibular schwannoma patients, and bring these in culture. And we do different kinds of studies with this. We can keep them for 6 months in culture with still hair cells being there. And this is an example of some studies in which we affected them with adenovirus to see whether gene therapy would be specifically targeting hair cells or not. Oh, somewhere. So that's actually the left part of this slide. So this is like really this human inner ear tissue we're using. And then we, of course, have the organoid model. And for you guys that are not really familiar with organoids, I put here the definition, and it's a miniaturized and simplified version of an organ produced in vitro in 3D that mimics the key functional, structural, and biological complexity of that organ. So for the inner ear, that means that we have like a 3D something growing that actually has some of the cell types we also have in our inner ear. And we make this actually from stem cells, from human pluripotent stem cells or human embryonic stem cells. And we're doing what we do is just mimicking human inner ear development. So if we look at this figure, it shows you how from the really early basilar stage of the human development, the inner ear actually develops. So it's from, you get ectoderm formation. So you get ectoderm on the outside. In a specific part of the ectoderm, you get like an area, which is called a placote domain. And this placote domain will give rise to the otic placote. This otic placote will invaginate, form a vesicle, and this vesicle will further mature to this whole inner ear that you actually know. And even neurons are actually derived from this otic vesicle. And at each of these steps, you have a lot of different signaling pathways that they are actually really important for the development. So you have TGF-beta, BMP signaling, FGF signaling, Wnt signaling that really contribute to the development of the coat and the inner ear. What we actually do is in the lab, we do the same thing. So we start out with these pluripotent stem cells. We treat them in the same way. So we are inhibiting signaling pathways and we're stimulating signaling pathways really good to go from milestone to milestone. So ectoderm is the coat domain, otic per coat. Then we get this otic vesicle, and this otic vesicle matures to the vesicle, which contains hair cells with actually neurons going there. And this is actually how an organoid looks after 75 days of culture. So it's a cross section, is a few millimeters in size, and there are a lot of different cell types in there. So it doesn't really have this really nice configuration we know from the inner ear. So you don't have a cochlea spiral shape, you don't have this semicircular canals, but at least a lot of these cell types we have in the inner ear are present. So if we zoom in, these vesicles actually contain the hair cells. And you can see here are the hair cells in yellow. You can see here all patches of hair cells being present. And if you look more closely and we do another staining, we can see that with the hair cells, we recognize even stereocilia. And you can see that we have neurons actually really protruding through these hair cells, making connections. So this is a model actually we're working. To get a better understanding how complex actually this organoid model is, we perform different studies using single cell RNA transcriptomics. I want to focus on one of them. And actually, we looked at a really late stage. So we had day 75 to day 110 organoids. We performed single nucleus and single cell RNA sequencing on this. I'm not sure how familiar actually with this method, but it means that with the organoid, we dissociate them to single cells. And then we can look per cell which genes are actually turned on. And because cell types differ in gene expression, you can get an idea of the cell types you have actually in there. And you can then visualize this in a 2D projection in which every dot is a cell. So you have here really small dots. And if they're really close together, they form like a cluster. And then we can look at these gene expression within these clusters. And then we can get an idea of actually the cell types that are present. So we came up with a lot of different cell types. And these are definitely not only inner ear cell types, because we also recognized endothelial cells, which actually made like vesicle-like structures in our organoids. We recognized swan cells, which actually really nicely surround neurons in our organoids. We have skeletal myocytes developing. That sometimes also that also explains why we sometimes saw these organoids actually contracting. So you could see like a ball which you have in culture actually contracting. We have melanocytes. Melanocytes are really important in our skin, but they also play a role in our inner ear. Now I'll come back to this later. And we have epidermal cells. So these are the cells that make up the choroid plexus and make your cerebrospinal fluid. But more importantly, we also identified, of course, these inner ear cell types in there. So we have hair cells, we have otic epithelial cells. So these are sensory cells, but also other cell types, and we have a specific population of otic mesenchyme in there. To get a better understanding actually how these otic cell types resemble the human inner ear, we also generated a small data set of 2 fetal stages of inner ear development and included an adult urtical ampule and also did single nucleus RNA sequencing on this. And then you get this atlas, so it contains both vestibular epithelium, cochlear epithelium, but also all mesenchymal associated cell types. And we use the package then, which is called Symphony, to actually see how organoid data from the previous slide actually match with this atlas we just generated. So we try to project our cells on this reference atlas. And this is how it looks if we focus on the mesenchymal cell type. So we identified the otic-specific mesenchymal cell types in our organoids. On the left, you can see how it actually looked in the inner ear. So you have the human inner ear. So you have mesenchyme that's more as a gene signature, which is more associated with epithelium associated mesenchyme. And you have mesenchyme that's more associated with the outside, so the otic capsule, which is capsule associated mesenchyme. If we then project our organoid mesenchyme on there, we can see that it only partially overlaps, so it mostly overlaps with the epithelium associated mesenchyme. can also, of course, confirm this with staining. So we did here staining for Wouter, which was one of the genes expressed within this mesenchyme. You can see surrounding vesicles with hair cells, you have these Wouter positive populations. We did the same approach also for the epithelium. So here you have separation for your vestibular epithelium and your cochlear epithelium. And if we project our organoid epithelium on there, so the other epithelium and organoids, it only overlaps with specific populations. So it's only a small representation of the human inner ear we actually have in there. However, we also just had to talk, of course, about genes that actually lead to hearing loss. So we looked also into protein expression of genes that are actually known to contribute to hearing loss of vestibular disorders. And you can see that these are actually nicely expressed if you look at parts of these vesicles. So making this model actually a potential model to mimic genetic hearing loss. The last comparison we did is the hair cells. So here we have the hair cells of the human inner ear with immature hair cells. So these were the fetal hair cells. And here the adults' hair cells, which are type 1 vestibular and type 2 vestibular hair cells. And you can see that the organoid hair cells mostly overlap with the immature hair cells. And we don't really see this distinction in type 1 and type 2. However, if we do look a little bit more closely, we can see some characteristics of type 1 versus type 2 hair cells. So type 1 hair cells have a really specific calyx-like synapse, while type 2 hair cells only have these bouton-like synapses. And with electron microscopy in hair cells in our organoids, we could actually see that we have these bouton-like synapses, but also some of these hair cells have these calyx-like synapses, showing that we have both type 1 and type 2 hair cells immature with hair cells in there. And we did also confirm this by immunohistochemistry. So the next step is actually to show you how we actually use these organoids also for disease modeling. So we have different projects going on. We focus on ototoxicity. We look at the different forms of genetic hearing loss, but also to a viral infection that actually contributes to hearing loss. And for this presentation, I would like to focus on two of the genetic forms we're working on. And that's actually Usher syndrome type 2 with mutations in the gene USH2A, which leads to congenital hearing loss, and it leads to the protein expression of usherin, which is expressed in the stereocilia of the hair cells. I just explained. And then we have the second one, which is deafness A9, and it's a non-syndromic form of hearing loss with more later adult onset, and that's the protein cochlein, which is expressed within this mesenchyme. And I can say this project is done by Esther and Winnie from our group, but also in collaboration with the Valk. If you look at these genes and look at how these are expressed actually in our organoid data, we can see that these are nicely expressed. So the USH2A gene is expressed in the hair cells in our organoids, and we have the cochlein gene, which is actually expressed within the optic mesenchymal cells. So confirming that we can use actually our organoids maybe to model these diseases. So we use the same approach for both the cell lines. So we have for both diseases. So we have cell lines that are actually derived from patients. We have on the bottom a healthy control cell line, so it's from a normal person. And then we also introduce the same mutation in this cell line as the patient cell line. So these two have the same genetic background. So we're using variation. If we look at the cell lines, we can see that we can generate organoids with all of them. So we still, for the USH2A, Usher type 2 organoids, we have hair cells in there and stereocilia. That's what we would expect. And for deafness A9, actually, you can see that we have these cochlein expression genes in the mesenchyme. So the next step is actually to see, can we see phenotype in there? So this is for USH2A. It means that in the healthy organoids, we have a band at this height. Only we know that because of the mutation, you get a longer RNA molecule. So that means that they will be higher. And that's also what we see. So both in the isogenic mutant in the patient line, we have longer RNA molecules. For the tetanus A9, we do a little bit a different approach because we look at the ratio between wild type and mutant transcripts. In a normal, healthy situation, you have a really high number. Only in organoids from 2 different days, you can see that it's really low. So that means that you have relatively more mutant present than wild type RNA. So this is just to establish that we have a working model of these 2 diseases. And the next step is actually to see whether we can restore these. So we're treating them with antisense oligonucleotides, so RNA therapeutics, which actually for H2A, the goal is to restore the function, and actually for tetanus A9 is to get rid of the bad RNA which is in there, so only the good RNA will stay there. So when we treat our organoids with fluorescent antisense oligonucleotides, you can see that these actually nicely enter our organoids, and we can also confirm this with an RNA scope. The tissue is a little bit damaged, but what you see here is that everything that turns more purple or pinkish is actually has taken up this anti-oligonucleotide protocol. So we're still analyzing a lot of the results, but these are some preliminary results from the tetanus A9 organoids which were treated with these therapeutics. And what you can see here is that if they are untreated or treated with scambot RNA therapeutics, in which you wouldn't expect any effect, the ratio is at this height. However, if we treat them with enough anti-oligonucleotides, you can see that this ratio changed a little bit. So this is like a first indication that actually we're treating our organoids, the tetanus A9 organoids. So then we're gonna go to the last part of the presentation. It's actually this next generation of innate organoids, like the full innate ear organoids we now make. I showed you this before. The reason I show it again is because its organoid models are incomplete, and specifically we are lacking these dark cells. And these dark cells are quite important because these can be seen as the power source of the inner ear. You can see here a schematic on the left of the cochlea, with the same holds true actually for the vestibular system, is that you have 3 major players in there. So you have your hair cells, which of course act as like the receiver cells, and they give your signal to the neurons, and the signal goes to the brain. But the system only works because we have the power source cells in there. So that's for the cochlea vascularis, and for the vestibular system, the dark cells. These actually make sure that the endolymph actually has a specific potassium concentration in there by which the hair cells can function. That's also the reason why if these power source cells are affected, for instance, in these genetic syndromes, it actually leads to hearing loss and also balance issues. But also if you look at more diseases, these triavascularis and these dark cells are actually affected. And just to show you, hypothyroidism is also in there. So we had to talk this morning, right? So maybe it has something to do with dark cells. We don't know. So to get a better understanding actually how these power source cells arise, there's not much known in literature about this. We generated a more extensive RNA transcriptomic atlas of early first trimester and up to early second trimester in the ears, really looking into how these cell types develop. So schematically, you can see that the cochlea undergoes like a huge change during this time window, in which we really have the triavascularis developing, but also, of course, the organ of corte with the hair cells. The vestibular system is already a little bit more advanced, but in this time period, you get type 2 versus type 1 specification, and you can see that these dark cells with melanocytes are actually developing in this time window. So what we did, we actually isolated nuclei from these inner ears and performed 10x genomics RNA sequencing. And after quality control and integration, we called the atlas the HYDRA we generated, and that stands for the Human in Ear Development Single Nucleus RNA Sequencing Atlas. And this is how it looks. So it has a lot of different cell types again, because like the whole membranous labyrinth was captured. So we have our cochlear epithelial cells, vestibular epithelial cells. We have a lot of mesenchymal populations, also some other capsule cells, but neurons here, hair cells here, endothelial cells, melanocytes, swan cells, immune cells. So really capturing like the really broad diversity of cell types we have in our TV here. So what we next did to actually focus on these power source cells, so dark cells and the lateral roof cells, which will become the area vascularis of the cochlea. So I show you now the results for this these lateral roof cells. So these become the area vascularis of the cochlea, and we can generate a mathematical timeline actually. So this is called pseudotime, which is the same in this plot and this plot. And we can look at genes that are actually known to be expressed really early on in these cells. So you can see that these expressions really high early on and low later. And we can look at genes that are actually known to be expressed later. Just to validate is that with this, we can identify two states, so an early state and a late state. We subsequently actually divided these two states in this early and late and looked at how these gene expression actually differs. So some of the bold expressed genes are actually known to be either specific early or either specifically expressed late. But this gives like an indication on how this early state differs from a late state. Next, we try to correlate these genes to more information. So what we did is a gene set enrichment analysis in which we looked specifically at signaling pathways, to get an understanding on which signaling pathways actually contribute to the development of these cells. And I just want to focus on the early state, and the top one hit was hedgehog signaling. So this gives information that hedgehog signaling actually might play a role in this really early development of these lateral roof cells in the cochlea. The interesting thing is if we do the exact same approach for the dark cells of the vestibular system, we can also recognize an early and late state. We do this separation in genes, and if we do the gene set enrichment, again, hedgehog signaling pops up in this early state. So that's why we decided to look a little bit more into this. We can look at all the different genes that are actually known to play a role in this signaling pathway. But for now, I just want to focus on the effectors. So these are the different GLIs. GLI1 is known as a major activator of this pathway, and GLI3 is known as a major repressor of this pathway. If you look at this, you can see that you see more expression of GLI3 compared to GLI1 in both the early state of the lateral roof cells and also the early state of the dark cells. So that means that probably hedgehog signaling is repressed during this really early development. So the next step is actually to validate this, and then actually where we use the organoid model for, because this organoid model lacked these dark cells. So we thought what would happen if we do, like at a specific time point, actually inhibit hedgehog signaling, which is actually help in developing these dark cells. And that seems to be the case. On the left, you can see here now like an organoid, which is like a normal situation with the marker we use were actually DAG1 for the dark cells. We don't see any DAG1 expression within the epithelium. However, if we treat them with this hedgehog inhibitor, we can see that these DAG1 cells actually really nicely develop. So we have presumable dark cells now in our organoid model. And the interesting thing is that the sensory part, so where the hair cells are, is still preserved. We can look a little bit more closely to other characteristics. So these first images are actually the melanocytes. So melanocytes are known actually to interact with this epithelium and really form connections and contribute to the function of these dark cells. And we can see that these melanocytes actually really home to this epithelium. Also disrupt the basal membrane, as is known from human inner ear development. And we can look at some proteins that are actually known to contribute to its function. So these are ion channels, pendrin, ATP1B2, and partin. And we can see also that these are expressed in these dark cell, the DAG1 positive cells. We can also look a little bit later. So the previous slide I was I think day 60 of organoids. So now we look at day 110 organoids, and we can see that these melanocytes, so this whole area actually was DAG1 positive. But if you zoom in on them, these melanocytes, they really have now projections towards the lumen. And this is like a one-to-one comparison also what we see happening in human inner ear development with first the melanocytes going there, but then also really going in the epithelium. And another protein we looked at was KC and Q1. So that's one of the more later proteins expressed in these cells, and that's also being expressed at the apical site. So a quick recap. So I showed you a little bit on what we did, what we're doing in Leiden. So we're working with inner ear organoids. We try to mimic diseases, more specifically USH2A and deafness A9 I showed you. And we also now have organoids with these dark cells, if you dark cells in there. So with that, I would like to thank everyone on this slide, especially all the members of our group, and have any questions.